Biomolecules. So what is meant by biomolecule? Bio means what? Living. Molecule means carbon made components. So carbon made components which is present in living system. Carbon made components which is present in the living organism is what we can call biomolecules. How many biomolecules are there in our body or in the living system? In the living system, how many different type of biomolecules are there? Four different type of biomolecules are there, students. Speak out what the four. Carbohydrates, proteins, lipids, nucleic acids. All are made carbon. Along with the carbon, other components also there, but carbohydrates, proteins, lipids and nucleic acids. But in these four biomolecules, come on, which biomolecule is helpful for inheritance? Nucleic acid. Other nucleic acid we are discussing today class, nucleic acid. Remember students, last year from molecular base of inheritance, 9 questions they are asked from molecular base of inheritance. 9 into 4, 36 marks illindane kelidru. I observed that one, okay na? So only molecular base of inheritance. But every year they are giving that one 5 to 6 questions from this. Okay na? But this year means what? Last year matra 9 questions they are asked. Okay, this one only indicating how much importance is there for this chapter. Okay na? So, but daily concept you have to finish that day itself only. Okay, textbook, read properly. Along with that one, I can give few extra points also. Okay, students. Okay. So nucleic acids. Why name is came that one nucleic acid? Come on, the nucleic acid and the word yaake bandide andre? Because it is present in the which one? Nucleus nallirutte. And why it is called acids andre? Because of acidic nature. Why they are having the acidic nature andre? Because of which one? Phosphoric acid. Correct? Phosphoric acid is present. It is present in nucleus, plus it is contain phosphoric acid. Phosphoric acid also present. Adike acidic nature. Got the clarity, students? Okay. Nucleic acids. Nucleic acids are first discovered by whom? Who first discovered that one, the nucleic acid? Friedrich Mischer. Remember, students, okay now? That is called Friedrich Mischer. Friedrich Mischer is first discovered that one, the nucleic acids. Friedrich Mischer. In 1869, they discovered that one, the nucleic acid. On nucleic acid nalli, which nucleic acid they are discovered? DNA. Why? Because nucleic acids, how many types are there? How many types of nucleic acids are there? 2 types. Why? Because deoxyribonucleic acid. So DNA, RNA both are together called nucleic acid, students. DNA and RNA both are together called nucleic acid. But in nucleic acid, DNA primarily present in, majoritarily present in the which part of the cell? Nucleus, correct? And little amount of the DNA also present in cytoplasm. Remember, students, where does, other than nucleus, where does DNA also present? Mitochondria and chloroplast also. Chloroplast also, circular DNA is present. Understood, students? Okay, no? So DNA. RNA. Where does RNA is present? RNA primarily present in cytoplasm. Little amount of RNA also present in nucleus, but primarily present in the cytoplasm. Correct? So both DNA and RNA both are called nucleic acid. Nucleic acid are first discovered by Friedrich Mischer. Okay, where it is discovered that one? In the pus cell. Pus. You already know that one. After the getting the injuries, come on, the pus formation is occur. Pus is which in color? Whitish in color. Pus is nothing but what? Dead WBC cells. Correct? Dead WBC cells. WBC cells are nucleated or enucleated? Nucleated. So nucleus is compulsory is present. Nucleus in the, he has separated that one of the chemical component, and what he has given the name? He given the name, okay na? He given the name, what is the name? Nuclein. Nuclein anta hesaritira. Very important, students, okay na? So nuclein, the word also given by Friedrich Mischer. But later who altered this name andre Altman. Altman is changed the name is nucleic acid. Nucleic acid anta hesaru change madidru yaru andre Altman. Why nuclein is changed into nucleic acid andre? Because nuclein is having which nature? Acidic nature. Just before I said why these nucleic acids are acidic nature andre? Because of which group? Because of which group? Phosphoric acid. Because of the phosphoric acid we can also call phosphate group antane helbodra. Phosphoric acid also called phosphate group because of the phosphate group and they are having the acidic nature. Clear student? This is the history of the nucleic acids. Okay. Next we can go for nucleic acids andre DNA ಬಗ್ಗೆ discuss ಮಾಡೋಣ. First nucleic acid is DNA. Deoxyribonucleic acid. So DNA, it is generally acts as a which one? Genetic material. It is generally acts as a genetic material ಅಂತ ಹೇಳ್ಬೋದು. Genetic material of all organism. It is a genetic material of all organism. Except which one? Viruses. Few viruses. Except few viruses. Can you speak out which viruses does not have the DNA? Yes, TMV virus is there, HIV is there. Remember students, HIV. Even coronavirus also completely it is having the RNA, etc. Plus pi. Okay, now X174 bacteriophage also. Sorry, it is having the DNA. Okay, so TMV, HIV and QB bacteriophage. QB bacteriophage. It is contained that one. RNA is a genetic material. Few viruses. Okay, now if few viruses nalli RNA is a genetic material. Okay, next one. So what is DNA? What is DNA? DNA is a genetic material. Anta helidandre adu correct alla. What is DNA? DNA is called is a polymer of which one? Polymer of yes, deoxyribonucleotides or nucleotides. It is a polymer of nucleotides. Or it is a polymer of, or one more definition also there. It is a polymer of many monomers. Come on, what are the monomers? Many monomers such as which one? Nucleotides. Nucleotides give one more name in the deoxyribonucleotides. So DNA is a polymer. Poly under in the many. So many nucleotides. Which type of nucleotides are there? Deoxyribonucleotides are there. Many deoxyribonucleotides. One link agi, they will form a chain. That's why it is a polymer of deoxyribonucleotides or polymer of many monomers such as deoxyribonucleotides. Get the clarity, students? Okay, now DNA. If DNA can be hydrolyzed, but one more important thing now. But all organisms, majority of the organisms, which one is the genetic material? DNA is a genetic material. It is consist of nucleotides. What is the name of the nucleotides now? Deoxyribonucleotides. And what is the name of the nucleotides we can observe in the RNA? Ribonucleotide, correct? Adralli ribonucleotides, idralli which one is there? Deoxyribonucleotides. But the deoxyribonucleotides or nucleotides number is same in all organisms DNA? No. But in your NCERT textbook, they are given crystal clear. That one you should remember, students. Okay, that one only I am giving. So first of all, bacteriophage lambda. Bacteriophage lambda is there, bacteriophage lambda. The bacteriophage lambda is consists of how many nucleotides or how many base pairs? 48,502 BPs are present. Base pairs are present. That is called Psi-X 174 bacteriophage is there. Bacteriophage is there. Idralli how many? Yes, 5,386. Yenidu BPs? No, nucleotides. Very important, students. Nucleotides. For your confirmation or confusion, they can make it to BPs and the So by this what you are understand? E bacteriophage nalli come on, they are having a single stranded DNA or a double stranded DNA? Single stranded DNA, ssDNA anta tilkoda. Very very important law. ssDNA, ss full form and single stranded DNA is there. Pi into 174 bacteriophage. Like after that one, okay, next one more also they are given, Escherichia coli. Come on, Escherichia coli nalli how many nucleo base pairs are there? 4.6 into 10 to the power of 6 BPs are present. 4.6 into 10 to the power of 6 BPs are present. Okay, next one, the haploid set of human genome. Haploid set of human genome is commonly consist of how many? 3.3 into 10 to the power of 9 BPs. So base pairs anta mention ಮಾಡ್ತಾ ಇದ್ದಾರೆ ಅಂದ್ರೆ ಅದು ಡಬಲ್ ಸ್ಟ್ರಾಂಡೆಡ್ DNA DSDNA ಅಂತ ಕರೀತಾರೆ. DSDNA, DS full form double stranded. Clear students? Haploid set of human genome. Haploid andre enda, that is how many chromosomes? 23 chromosomes. 23 chromosomes andre one set chromosome if you are completely observed 3.3 into 10 to the power of 9 BPs. Adu diploid anta takshana 6.6 into 10 to the power of 9 BPs that. ಅರ್ಥ ಆಯ್ತಾ ಸ್ಟೂಡೆಂಟ್ಸ್? ಇದು ಸಿಂಗಲ್ ಅಂದರೆ ಓನ್ಲಿ ಒನ್ ಸೆಟ್ ಮಾತ್ರ, ಯಾಕಂದರೆ ವಿ ಹ್ಯಾವ್ ದ 2 sets ಆಫ್ ಕ್ರೋಮೋಸೋಮ್. 23 one set, 23 another set, both are called homologous chromosomes ತಾನೆ. ಈ 23 set is came from the mother and remaining 23 came from father. ಅಲ್ವಾ? We have that one, the both our mother father chromosomes we have. Mother ಇಂದ ಬಂದಿದೆ one set, father ಇಂದ ಬಂದಿದೆ one set chromosomes we have. ಅದಕ್ಕೆ haploid set of chromosomes ನಲ್ಲಿ 3.3 into 10 to the power of 9 BPs are there. ಅದೇ diploid set ಅಂದರೆ ಅದಕ್ಕೆ ಡಬಲ್ ಮಾಡ್ಕೊಳ್ಳಿ 6.6 into 10 to the power of 9 BPs. Get the clarity students? But this one maybe they can ask for either in the statement type of question or either they can give in the match the following question. Got the students? Okay, now 5386 nucleotides raw. That is called pi x 174 bacteriophage. Okay, bacteriophage lambda nalli matra 48502, E. coli nalli matra 4.6 into 10 to the power of 6 BPs raw. 9 anta kodtare, wrong adu, 4.6 into 10 to the power of 6 base pairs only. Idunu ashtira, adike specific ಆಗಿ ಓದ್ಕೊಂಡು ಹೋಗಿರಾ when you are reading that. Clear students? Okay, so after this one we can go for in detail information of DNA. DNA if you are completely hydrolyzed, partially hydrolyzed ಮಾಡಿದಂದ್ರೆ ಓಕೆನಾ, okay, it is completely converted into which one? Nucleotides. Partially if you are hydrolyzed ಮಾಡಿದಂದ್ರೆ nucleotides ಗೆ ಡಿವೈಡ್ ಆಗುತ್ತೆ, ಮತ್ತೆ nucleotides ನಿ ಮತ್ತೆ partially hydrolysis ಮಾಡಿದಂದ್ರೆ ಏನಾಗುತ್ತೀರಾ? Partially hydrolyzed ಮಾಡಿದಂದ್ರೆ ಮತ್ತೆ it is converted into which one? Nucleosides and phosphates. Nucleosides and phosphate ಗೆ ಕನ್ವರ್ಟ್ ಆಗುತ್ತಾರೆ. Clear students? Okay na? So what is meant by nucleoside? Nucleoside andre that is called, yes, nitrogen base plus which one? Sugar. Direct here only I am writing now. This is about the nitrogenous bases and sugar. That is called pentose sugar is present. Mainly pentose sugar is present. Okay, e nitrogenous bases again we are divided into how many types now? That is called 2 types are nitrogenous bases are there, that is called purines and pyrimidines. Speak out students, which one is called purines? That is called adenine and guanine both are called them as purines anta karitira. Okay na? ಈಗ pyrimidines andre which one? Thymine, cytosine and uracil, but uracil is present only in RNA. Got the clarity students? Okay now, so DNA is the polymer of nucleotides already know. Adike if you are hydrolyzed madidandre, hydrolysis andre meaning enna? In the presence of water, if the DNA become divide madidandre, come on, what is the name of the enzyme is helpful for digestion of the yes, what is the name? DNase. That's why DNase is converting the DNA into which one? Nucleotides. ಆ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಗೆ ಒಂದು ಸ್ಪೆಸಿಫಿಕ್ ನೇಮ್ ಹೇಳಿದಾರ, ಏನದು? ಡಿಆಕ್ಸಿರೈಬೋನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಅಂತ ಹೇಳ್ಬೋದ್ರ, RD ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಅಂತ ಹೇಳ್ಬೋದು. ಡಿಆಕ್ಸಿರೈಬೋನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್. ಈ ಡಿಆಕ್ಸಿರೈಬೋನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಮತ್ತೆ ಹೈಡ್ರಲೈಸ್ ಮಾಡಿದಂದ್ರೆ ಕಮಾಂಡರ್ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ಸ್ ಅಂಡ್ ಫಾಸ್ಫೇಟ್ ಗ್ರೂಪ್ ಈಸ್ ಫಾರ್ಮ್ಡ್. ಸೊ ಅಂದ್ರೆ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಪ್ಲಸ್ ಫಾಸ್ಫೇಟ್ ಬೋತ್ ಆರ್ ಟುಗೆದರ್ ಕಾಲ್ ವಿಚ್ ಒನ್? ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್. ಕರೆಕ್ಟಾ? ಓಕೆನಾ? So next one, nucleosides, only two components are there, that is called nitrogenous bases and pentose sugar. Nitrogenous base and pentose sugar. Nitrogenous bases are divided into two types, that is called purines and pyrimidines. Later we can discuss in detail of every concept. ಒಂದೊಂದು ಕಾನ್ಸೆಪ್ಟ್ ಬಗ್ಗೆ ಇನ್ ಡೀಟೇಲ್ ಆಗಿ ಹೋಗೋಣ. ಫಸ್ಟ್ ನೈಟ್ರೋಜನ್ ಬೇಸಿಂದನೇ ಶುರು ಮಾಡೋಣ. ಅರ್ಥ ಆಯ್ತಾ ಸ್ಟೂಡೆಂಟ್ಸ್? ಓಕೆನಾ? ಇಂಟ್ರಡಕ್ಷನ್ ಅರ್ಥ ಆಯ್ತಪ್ಪ? ಓಕೆನಾ? ಫೆಡ್ರಿಕ್ ಮಿಷರ್ ಈಸ್ ಫಸ್ಟ್ ಡಿಸ್ಕವರ್ಡ್ ದಟ್ ಆನ್ ದ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್. ಓಕೆನಾ? ಇನ್ ದ ಪಸ್ಸೆಲ್ in the 1869, year also ನೆನಪಿಟ್ಕೊಳ್ಳ ಸ್ಟೂಡೆಂಟ್ಸ್, year also you should remember, and he has given the name nuclein. Next, what is DNA andre? It is a polymer of deoxyribonucleotides, or it is also consist of the polymer of many, which one are monomers. What is the name of the monomers? Deoxyribo, which one? Nucleotides. Then we can go for which one? Nitrogenous bases. What we can discuss? Nitrogenous bases. We can go for the discussion. Okay na? Era again you want to repeat? No. Okay. So important concept ಮಾತ್ರ repeat ಮಾಡ್ಕೊಂಡು ಹೋಗ್ತೀನಿ. Okay. ಈಗ next ಇದೇ DNA ನಲ್ಲಿ nitrogenous bases we have to go in detail. Okay na? So DNA ನಲ್ಲಿ nitrogenous bases. Nitrogenous bases. Nitrogenous bases. In nitrogenous bases only, first of all, we can discuss about purines and pyrimidines. Purines and pyrimidines ಬಗ್ಗೆ in detail ಆಗೋಗೋಣ. Nitrogenous bases, purines and pyrimidines. What are purines? Purines are commonly, it is a dicyclic compounds. Dicyclic compounds ಅಂತ ಹೇಳ್ತೀವಿ. Purines are dicyclic compounds. Pyrimidines are which one? Monocyclic compounds. Monocyclic compounds. Okay. Why sir it is called dicyclic andre? I am drawing the diagram of this. Okay. Dicyclic compounds. ಈ dicyclic compounds ನಲ್ಲಿ you see in the first position ಆಗ್ಬೋದು, in the third position ಆಗ್ಬೋದು, and seventh position ಆಗ್ಬೋದು, ninth position ಆಗ್ಬೋದು. Which one is present? Nitrogen is present. First, second, third, fourth, fifth, sixth, seventh, eighth, ninth. ಇವೆಲ್ಲ dicyclic compounds. First point important students. Second one is called the mass. Okay na? Nitrogen present at which position? First, third, seventh, ninth position. Ninth position. This point also you should remember. Okay, next. Purines attached to C1 position of sugar. C1 position of sugar by its ninth position. This point also very very important. Purines attached to C1 position of sugar, ಅಂದರೆ ಫಸ್ಟ್ ಪೊಸಿಷನ್ ಆಫ್ ದಿ ಶುಗರ್ ಬೈ ಇಟ್ಸ್ ನೈನ್ತ್ ಪೊಸಿಷನ್. ಇದು ಸ್ವಲ್ಪ ಅರ್ಥ ಆಗಬೇಕಂದ್ರೆ ಸ್ವಲ್ಪ ಟೈಮ್ ಆಗುತ್ತರ. ಅಂದರೆ ಸೊ ಫಸ್ಟ್ ಮಾತ್ರ ಪಾಯಿಂಟ್ಸ್ ಕೊಡ್ತಾ ಇದ್ದೀನಿ, ಆಮೇಲೆ ಡಿಟೇಲ್ ಆಗಿ ಒನ್ ಬೈ ಒನ್ ಐ ಕ್ಯಾನ್ ಗಿವ್. ಈಗ ಮೋನೋಸೈಕ್ಲಿಕ್ ಕಾಂಪೌಂಡ್ಸ್. ವೈ ಇಟ್ ಈಸ್ ಕಾಲ್ ಮೋನೋಸೈಕ್ಲಿಕ್ ಅಂದರೆ ಓನ್ಲಿ ಒನ್ ಸೈಕಲ್ ಓನ್ಲಿ ಪ್ರೆಸೆಂಟ್, ಬಟ್ ಹಿಯರ್ ಆಲ್ಸೋ ನೈಟ್ರೋಜನ್ ಈಸ್ ಪ್ರೆಸೆಂಟ್. ಮಿಕ್ಕಿದೆಲ್ಲ ಪಾರ್ಟ್ನಲ್ಲಿ ವಿಚ್ ಒನ್ ಈಸ್ ಪ್ರೆಸೆಂಟಾ? Here nitrogen is present andre in all situation also nitrogen or carbon? Carbon is commonly present. That's why I am not mentioning there. Okay na? It is about the normal diagram of the purines, that is. So first, third, seventh, ninth position which one is there? Nitrogen is present. But here nitrogen present at which position? At which position? First and third position. This point important. Next, one more important point is there. Ena important point? Yes, pyrimidines attached C1 position of sugar by its third position. That is important students. Okay, so purines and pyrimidines, these are about the important points. First carbon of the sugar can be bound with the 9th position by its 9th position, so here 3rd position. Okay, now you have to speak out, and purines, which one is called purines? Adenine and guanine. Adenine and guanine both are together called purines. You get thymine, cytosine. Thymine and cytosine both are together called which one? Pyrimidines. Okay, now I can give the diagram of every atomic, every nitrogenous base atomic structure I can give now. Okay, na? Okay, are you understand this tabular column, students? Tabular column artha aita? Any doubts idya? Any doubts idya? Yes, easy agidalla. DNA is hydrolyzed madidandre nucleotides. Matte nucleotides nalli what are the components are there? Nucleosides plus phosphate. Nucleosides is made up of which one? Nitrogenous base and pentose sugar. Aa nitrogenous bases baggene now in detail agi discuss ಮಾಡ್ತಾ ಇದ್ವಿ. Okay, so now I can give the atomic structure. Why? Because before last year they are asked one question from these nitrogenous bases. Nitrogenous bases indane okay na questions kelidru. Ade first adenine diagram, adenine atomic structure I am writing now. This about the adenine dicyclic compound. Ee dicyclic compounds nalli here ammonia group is there in the 6th position, and here nitrogen is there, here also nitrogen, here also nitrogen, here also nitrogen is there. Okay na? So now this is turning. Idu turn ಮಾಡಿ ಬಾ, ಏನು ಪ್ರಾಬ್ಲಮ್? ಮತ್ತೆ okay, so this is about the adenine. You are not writing anything. I will check your notes. Write atomic structure also you have to draw. Don't turn the page. Okay, so this is about adenine. Next one is guanine. Guanine. So here I am writing about the guanine. So this is guanine. Guanine, it is contain same nitrogen, same now here also nitrogen, but here H is present. Okay, next here also, this is called guanine. Both are called purines. Again, next one the pyrimidines. Pyrimidines nalli now I am writing about, this is thymine. This is about the thymine. Now this is about the thymine. Okay, next one I am writing about after the thymine. Okay, cytosine. I am writing. Okay, so nitrogen, so here NH is present. Okay, so this is called cytosine. Okay, next I am writing that one uracil also. I am drawing about here. Okay, uracil also I am drawing same. But what is the difference between the uracil and thymine? You only speak out. Okay, no? Yes. They are asked about uracil. Okay, no? So these are about nitrogenous bases atomic structures I drawn on the board. Adenine, guanine, both are called purines. Thymine, cytosine, uracil, these three are called pyrimidines. That's why I written in one row. Okay, so here adenine, guanine, pyrimidines and dicyclic compounds. Thymine, cytosine, uracil is called which one? Monocyclic compounds. Andre only one circle only present here. Monocyclic compounds. Okay, then yen sir ಈ ಪಾಯಿಂಟ್ Second point I written, okay. Now first point we got it, sir. First, third, seventh, ninth positionally nitrogen is a here, okay. Nitrogen is present in first and third carbon or third position. Third position it is there, okay. But before last year NEET question ಏನು ಗೊತ್ತಾ? thymine uracil. ಏನು difference ra? Same here also. Yes, methyl group is present. Methyl group is present in the which carbon? Yes, which carbon now? Sorry. This is about the fourth, correct? Fifth and sixth. Which carbon mele methyl group is there? Yes. Okay, now which carbon is contained methyl group? Fifth carbon. But other methyl group is we can observe in the uracil. No, we can't observe in the uracil. Okay, now we can't observe the methyl group in uracil. Other methyl group, okay? Uracil ke methyl group, if you are add that one, come on, this one also convert into which one? Thymine. So that's why thymine ge 5-methyl uracil anta bariboda. Yes, 5-methyl uracil. Igo idera kottidu. The following nitrogenous base is called 5-methyl uracil. Yes, which one is called 5-methyl uracil? Thymine. Clear, students? But atomic structures ಏನು ಕೊಡಲ್ಲ ಜನರಲಿ. ಬಟ್ ನಿಮ್ಮ ಬಯೋಮಾಲಿಕ್ಯೂಲ್ಸ್ ಚಾಪ್ಟರ್ನಲ್ಲಿ ಮಾತ್ರ ಇದು ಕೊಟ್ಟಿದ್ರು, ಥೈಮಿನ್ ಕೊಟ್ಟಿದ್ರು ಪ್ಲಸ್ ಯುರಾಸಿಲ್ ಕೊಟ್ಟಿದ್ರು. ಮಿಕ್ಕಿದೆಲ್ಲ ಅಟಾಮಿಕ್ ಸ್ಟ್ರಕ್ಚರ್ಸು ಅಷ್ಟು ಕೊಟ್ಟಿಲ್ಲ. ಬಟ್ ಅದಕ್ಕೆ ಬಿಡಬೇಡಿ ಅಂತ. ಸೊ ಥೈಮಿನ್ ಆ್ಯಂಡ್ ಯುರಾಸಿಲ್ ಬೋತ್ ಆರ್ ಹ್ಯಾವಿಂಗ್ ಸೇಮ್ ಅಟಾಮಿಕ್ ಸ್ಟ್ರಕ್ಚರ್ ದೇ ಹ್ಯಾವ್. ಬಟ್ ಫಿಫ್ತ್ ಪೊಸಿಷನಲ್ಲಿ ಮೀಥೈಲ್ ಗ್ರೂಪ್ ಈಸ್ ಪ್ರೆಸೆಂಟ್, ಬಟ್ ಹಿಯರ್ ಮೀಥೈಲ್ ಗ್ರೂಪ್ ಈಸ್ ಆಬ್ಸೆಂಟ್ ಇನ್ ದ ಯುರಾಸಿಲ್. Clear, friends? Okay. Again now. Okay. So nitrogenous bases are over. Second point artha agಬೇಕಂದ್ರೆ ಸ್ವಲ್ಪ ಟೈಮ್ ಆಗಬೇಕು. Okay. So now this is about position ಗೊತ್ತಾಯ್ತು ತಾನೆ? What is about the first? This is about the first position, second, third, fourth, fifth, sixth, seventh, eighth, ninth. ಇದು ninth position. ಇದನ್ನು ninth position. Okay na? But here which position is this one? Third position. Here also third position. Here also third position. Clear? Okay, now we can discuss after the nitrogenous base we have to discuss about the next concept in the that is called sugars. Alva nucleoside under meaning and what is meant by nucleoside meaning yes, nitrogenous base plus sugar. Nitrogenous base ಬಗ್ಗೆ okay idea ಬಂದಿದೆ. ಈಗ sugar ಬಗ್ಗೆ discuss ಮಾಡೋಣ. Okay, sugar, what type of sugar that is called pentose sugar. ಈ ಪೆಂಟೋಸ್ ಶುಗರ್ ಈಸ್ ವಿಚ್ ಟೈಪ್ ಆಫ್ ಪೆಂಟೋಸ್ ಶುಗರ್ ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಡಿಆಕ್ಸಿರೈಬೋಸ್ ಶುಗರ್ ಅಂತ ಕೇಳಿದ್ರ. ಡಿಆಕ್ಸಿರೈಬೋಸ್ ಶುಗರ್ ಈಸ್ ಕಾಮನ್ಲಿ ಪ್ರೆಸೆಂಟ್. ಓಕೆ, ವಾಟ್ ಇಸ್ ದ ಫಾರ್ಮುಲಾ ಆಫ್ ಡಿಆಕ್ಸಿರೈಬೋಸ್ ಶುಗರ್? C5H10O4. C5H10O4, that is about the formula of the deoxyribose sugar. Same atomic structure I am drawing again. This is about the atomic structure. Okay, so H or OH, H. Here also H. Okay, same here also H, and this is called CH2OH. Idra sugar molecules. Which sugar molecule idu? Deoxyribose sugar. This is called deoxyribose sugar. Again, next one more sugar also I am drawing. That is about the one more sugar diagram. What is the name of this sugar? This sugar is called as ribose sugar. This is called ribose sugar. In ribose sugar nalli, are you observing, students? Okay, no? Same here also OH is there, H is there, but here H, OH is present, H, OH, and here H and CH2OH. This is about the ribose sugar diagram, students. But ribose sugar formula in the C5H10O5. Oxygen ಐದು ಇರುತ್ತೆ ribose sugar ನಲ್ಲಿ. So C5H10O5, it is about the empirical formula of ribose sugar, students. Okay na? I think in biomolecules also ಈ concept ಎಲ್ಲ ಬಂತಾ? Biomolecules of your chemistry. ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ನಲ್ಲಿ ಸ್ಟ್ರಕ್ಚರ್ಸ್ ಎಲ್ಲ ಬಂತು ತಾನೆ? ಎಸ್. ಓಕೆನಾ? ಅಂದರೆ ಸ್ವಲ್ಪ ನಾನು ಫಾಸ್ಟಿಗೆ ಮೂವ್ ಆಗ್ಬೋದಾ? ಲಿಟಲ್ ಫಾಸ್ಟಿಗೆ ಮೂವ್ ಆಗ್ಬೋದಾ? ಓಕೆ, ಎಸ್. ಶುಗರ್ಸ್ ಆರ್ ಕಾಮನ್ಲಿ ವಿಚ್ ಶುಗರ್ಸ್ ಆರ್ ದೇರ? ಪೆಂಟೋಸ್ ಶುಗರ್. ಪೆಂಟಾ ಅಂದ್ರೆ 5 ಕಾರ್ಬನ್ಸ್ ಆರ್ ದೇರ. 1 2 3 4 5. ಅರ್ಥ ಆಯ್ತಾ? ಓಕೆನಾ? ಸೊ ಹಿಯರ್ ದಿಸ್ ಇಸ್ ಅಬೌಟ್ ದ ಫಸ್ಟ್ ಪೊಸಿಷನ್, ದಿಸ್ ಅಬೌಟ್ ದ ಸೆಕೆಂಡ್ ಪೊಸಿಷನ್, ಥರ್ಡ್ ಪೊಸಿಷನ್, ಫೋರ್ತ್ ಪೊಸಿಷನ್, ಫಿಫ್ತ್ ಪೊಸಿಷನ್. Okay, so 5 carbons are there. Are you observing, students? The sugars, which type of sugar is present here? Deoxyribose sugar is present. Deoxyribose sugar formula is C5H10O4. How many oxygens are there? Okay, now 4 oxygens only is there. 1, 2. Okay, now one more oxygen is there. 3 and 4. 4 oxygens only present. But carbons matra how many is there? 5 carbons. ಈಗ hydrogens ಮಾತ್ರ 10 ಆಗಿರುತ್ತೆ. ನೀವು ಕೌಂಟ್ ಮಾಡಿದ್ರೆ 10 hydrogens ಬರುತ್ತೆ. ಈಗ next one ribose sugar. Ribose sugar ನಲ್ಲಿ are observing, students? First position, second position. Second position ನಲ್ಲಿ which group is there? OH group is there. But here second position ನಲ್ಲಿ which group is there? Hydrogen group is there. Correct? So OH group ಇರುವಾಗ one oxygen is more or less? More. Okay na? So that's why what is the ribose sugar formula? C5H10O5. Okay na? So what is meant by deoxy? Deoxy meaning enna? One oxygen is less. Deoxy endrinda less. How many oxygens are less compared to that one, the ribose sugar? One oxygen is less. That's why what is the empirical formula of deoxyribose sugar? C5H10O4, and empirical formula is C5H10O5. O5 is the empirical formula. Okay na? So this is the nucleoside. Nucleoside nilade 2 components complete aitu. Nucleosides nilade 2 components. 2 components are finished. What are the 2 components? Nitrogenous bases are finished and sugar. ಈಗ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಹೆಂಗೆ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೋ we have to discuss on. Okay, but ಈ ಸೆಂಟೆನ್ಸ್ ಹಂಗೆ ಇರ್ತೀನೋ, ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಹೆಂಗೆ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ. This sentence. Next, write down nucleoside. Nucleoside. You already know nitrogenous bases, correct? Nitrogenous base plus sugar, both are called them as which one? Nucleoside anta karidira. Nucleoside. Okay, just before I said that one, this is called purines. Purines and pyrimidines, adenine, guanine, both are called them as adike ille inge barkora A, G anta barkori, thymine nalli T, C anta barkori. Okay na? Even uracil also barkori, that is called, but it is present in RNA. So nitrogenous base plus sugar is called them as nucleoside. Nucleoside henge formation agutandre, you see, this is about the sugar molecule. This is CH2OH, CH2OH, and this is about the first carbon is present here, first carbon. Okay, same H is present, OH is present, and this is about the H. So now this is about the sugar. Sugar molecule ge come on now purines attach ಆಗಬೇಕು. Purines are completely attached to that one, the sugar molecules, with the which position? The first position or second position? First position. This is about the first position of the carbon. No, first position of the carbon, you see, it is attached to the C1 position. C1 means what? First portion of the sugar by its which position? Ninth position. Okay na? Suppose here only I am complete drawing about the one of the nucleoside, one of the nitrogenous bases I am drawing. Okay, in nitrogenous basis nalli, yes, I am complete drawing like this one. So this is about the nitrogenous base. Okay, in nitrogenous base, are you remember, students? This is about the first carbon, and the second, third, fourth, fifth, sixth, seventh, eighth, ninth. Ninth position, what is there? Ninth position, what is there here? Nitrogen is there. Correct? So our nitrogen jotege. Okay, and here also nitrogen is there, but e nitrogen alli come on. Okay, ninth position of nitrogen ge plus first carbon e madhya. Okay, so one of the bond formation is occurs. What is the name of the bond? What is the name of this bond? Yes, glycosidic bond anta karidira. Important, students. Okay, sugar molecule, they can bind to the purines with their ninth carbon. Okay na? So first position of the carbon, first position of the sugar, ninth position of the which one? The purine. Ninth position of the purine, they can bind with each other by one of the bonds are present. What is the name? Glycosidic. Glycosidic bonds are present. Glycosidic bonds are present. The glycosidic bond is formed between the sugar and base, nitrogenous base. Same ade tarahara here also sugar jotege and purines can be bind. Sorry, pyrimidines can be bind. This is about the one of the sugar. This is about the one of the sugar. Okay, CH2OH. Same here also OH is present. Idralli, what is the name of this? You go here, This is called NH is there. Here also one of the bond is completely, water molecule is completely move. Water molecule move agi. What is the name of the bond is formed? Glycosidic bond here also formed. But glycosidic bond is formed in the which position? The yes, that is called which position of the sugar? Which position of the nitrogenous base? Third position. Third position. ಇದು ರೇರ್ ಆಗಿ ಹೇಳ್ಬೋದು ಸ್ಟೂಡೆಂಟ್ಸ್, ಓಕೆನಾ. ಸಿಂಪಲ್, ಇದರಲ್ಲಿ ಏನು ಜಾಸ್ತಿ ಏನು ಕ್ರಿಟಿಕಲ್ ಏನು ಕಂಡೀಷನ್ ಇಲ್ಲ, ಓಕೆನಾ. ಒನ್ಸ್ ಅಗೈನ್ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಸೊ ಪ್ಯೂರಿನ್ಸ್, ದಿಸ್ ಅಬೌಟ್ ದ ಪ್ಯೂರಿನ್. ಪ್ಯೂರಿನ್ಸ್ ಆರ್ ಡೈಸೈಕ್ಲಿಕ್ ಆರ್ ಮೊನೋಸೈಕ್ಲಿಕ್? ಡೈಸೈಕ್ಲಿಕ್, ಕರೆಕ್ಟಾ. ಈ ಡೈಸೈಕ್ಲಿಕ್ ಕಾಂಪೌಂಡ್ಸ್ is completely bind with that one, the sugar molecule with its ninth position of the purines first carbon of the sugar. Ninth position of the purine first carbon of the sugar with the help of which bond? Adhe tarha, okay na. Here also sugar is there. Sugar can be bind with that one, the pyrimidines with their first carbon as usual is there. First carbon jotige, okay na. Which position of the pyrimidine? Third position of the sugar. Same here also glycosidic bond is there. Here also glycosidic bond is there. Glycosidic bond formation is occurs by here also. Correct students, okay na. So why? Because here hydrogen is there. Here also hydrogen is there. Alva, hydrogen is there. Hydrogen is there. Same here hydrogen is there, like this one. Here hydrogen is there, here also OH is there, you know. Iveradu the hydrogen and OH, they are removed in the form of which one? The dehydration agi glycosidic bond is formed. Correct aitane? Ide aitane ninth position of purines. Ninth position of purines can be bind with the first carbon of the sugar by glycosidic bond. Then only which one is formed? Nucleoside formation ಆಗಬೇಕಂದ್ರೆ ನೈಟ್ರೋಜನಸ್ ಬೇಸ್ ಶುಗರ್ ಎರಡು ಇರಬೇಕು, ದೆನ್ ಓನ್ಲಿ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಈಸ್ ಫಾರ್ಮ್ಡ್. ಈಗ ಅರ್ಥ ಆಯ್ತಾ ಸ್ಟೂಡೆಂಟ್ಸ್? ಈ ಪಾಯಿಂಟ್ ಅರ್ಥ ಆಯ್ತಾ? ಓಕೆನಾ. ಸೊ ದಟ್ ಈಸ್ ಇಂಪಾರ್ಟೆಂಟ್. ಓಕೆ. ಸೊ ಗ್ಲೈಕೋಸಿಡಿಕ್ ಬಾಂಡ್ ಫಾರ್ಮೇಷನ್ ಅಲ್ಲಿ ಡಿಹೈಡ್ರೇಷನ್ ಅಕರ್ಸ್. ಡಿಹೈಡ್ರೇಷನ್ ಮೀನಿಂಗ್ ರಿಮೂವಲ್ ಆಫ್ ವಾಟರ್ ಮಾಲಿಕ್ಯೂಲ್ ರಿಮೂವಲ್ ಆಗಿ ಗ್ಲೈಕೋಸಿಡಿಕ್ ಬಾಂಡ್ ಈಸ್ ಫಾರ್ಮ್ಡ್. ಬಟ್ ಇಟ್ ಈಸ್ ಫಾರ್ಮ್ಡ್ ಇನ್ ದ nitrogen of the purines and nitrogen of the pyrimidine with sugar. ಅದಕ್ಕೆ ಈ ಗ್ಲೈಕೋಸಿಡಿಕ್ ಬಾಂಡ್ಗೆ ಒನ್ ಮೋರ್ ನೇಮ್ ಇಂದ ಎನ್ ಗ್ಲೈಕೋಸಿಡಿಕ್ ಬಾಂಡ್ ಅಂತ ಕರೀತೀರ. ಎನ್ ಗ್ಲೈಕೋಸಿಡಿಕ್ ಬಾಂಡ್, ಯಾಕೆಂದರೆ ಪ್ಯೂರಿನ್ಸ್ ನೈಟ್ರೋಜನ್ ಇದೆ ನೋಡಿ, ನೈನ್ತ್ ಪೊಸಿಷನಲ್ಲಿ ನೈಟ್ರೋಜನ್ ಇದೆ ಅಲ್ವಾ. ಸೇಮ್ ಥರ್ಡ್ ಪೊಸಿಷನಲ್ಲಿ ಆಲ್ಸೋ ನೈಟ್ರೋಜನ್ ಇದೆ ಅಲ್ವಾ. ನೈಟ್ರೋಜನ್ ಜೊತೆಗೆ ಬೈಂಡ್ ಆಗ್ತಾ ಇದೆ, ಅದಕ್ಕೆ ಈ ಬಾಂಡ್ಗೆ ಏನು ಹೆಸರು? ಎನ್ ಗ್ಲೈಕೋಸಿಡಿಕ್ ಬಾಂಡ್ ಅಂತ ಕರೀತೀರ. ಎನ್ ಗ್ಲೈಕೋಸಿಡಿಕ್ ಬಾಂಡ್. Okay, now, okay, we got that one, the clarity. Sir, nucleoside antakshna, yes sir, nitrogen based sugar is called nucleoside. Okay, but ega nitrogenous base. What is the name of the nitrogenous base? Adenine plus sugar. Come on now, adenine plus sugar, both are together what we can call adenosine anta karitara. Okay, next one. After that one more, yes one. Next one is called guanosine. Next one in the thymidine. Last one in the cytidine that. Now you have to speak out guanosine nalli what are the components are there? Guanine plus sugar, correct? Thymidine nalli which one is there? Thymine plus sugar. Thymine plus sugar is present, and cytosine plus sugar is what we can call cytidine. Got the clarity students? Okay na. So totally nucleosides, how many different type of nucleosides are there? 4 different type of nucleosides are there in the DNA. Clear? Idu jotege that is called the as uracil plus sugar molecule, what we can call uridine. But uridine is present only in RNA. RNA nalli matra uridine is present. Okay, in your examination what type of questions kelbodu andre, what are the components are there in the adenosine? Come on, adenosine nalli what are the components are there? Adenine plus sugar, like that one they can give. Okay na. Guanosine nalli guanine plus sugar, thymidine nalli thymine plus sugar, cytidine nalli ra, yes, okay na, that is called cytosine plus sugar is present in the cytidine. Okay ra, idra totally nucleosides ಬಗ್ಗೆ war. Okay, so what are the components of nucleoside? What are the components of the nucleoside? Nitrogenous base and sugar. Nitrogenous base and sugar both are together called what? Nucleoside. How many different types of nucleosides are present in the DNA? 4. away ಈ 4 nucleosides. Okay, now what we can discuss now, nucleotide ಬಗ್ಗೆ ಡಿಸ್ಕಸ್ ಮಾಡೋಣ. ಯಾರ ಅರ್ಥ ಆಗ್ತಿದೆಯಾ? Are you understand? Can I move that one? Okay, now next one is called nucleotide. So what is mean by nucleotide? Okay, now so nucleotide meaning in the nucleoside plus phosphate. Okay, now so phosphate group ಬಗ್ಗೆ ಫಸ್ಟ್ ಒಂದು ಎರಡು ಮಾತ್ರ. Phosphate group. Phosphate group. So phosphate group is, what is the formula of phosphate group? H3PO4 minus. H3PO4 minus andre phosphate group. They're having how many hydroxyl groups are there? Do you know? 3 hydroxyl groups are there, students. This H3PO4 minus empirical formula or diagram. This is about the atomic structure of the H3PO4 minus. Nodi, hydrogens are 3 are there, oxygens are 4 are there. Okay, na? So that is, so phosphate group H3PO4 minus, but idರಲ್ಲಿ okay na, paid electrons are there, ಅದಕ್ಕೆ minus ಅಂತ ಮೆನ್ಷನ್ ಮಾಡ್ತಾ ಇದೀವಿ, H3PO4 minus because of phosphate group, or it is also called which group ಅಂತ ಕರಿತೀರಾ? Phosphoric acid ಅಂತ ಕರಿತೀರಾ. actually phosphate group ನೇ one more name ಇಂದ phosphoric acid, it is called phosphoric acid. But the phosphoric acid present only in phosphoric acid present only in which one? Nucleotides nalli matra phosphoric acid is present, students. Okay, let us, we can discuss about types of nucleotides present in the DNA. Okay, nucleotides meaning in the nucleoside plus phosphate is called the mass. ಯಾಕಂದರೆ ಇವೆಲ್ಲ ಬೇಸಿಕ್ಸ್ ನಿಮ್ಗೆ ಅರ್ಥ ಆಯ್ತು ಅಂದ್ರೆ ಮಾತ್ರ ನೆಕ್ಸ್ಟ್ ಪಾಲಿನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಬಗ್ಗೆ ಹೇಳುವಾಗ ಈಸಿ ಆಗುತ್ತರ. ಓಕೆನಾ? ಈಗ ನೆಕ್ಸ್ಟ್ ಈ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ನಲ್ಲಿ ಫಸ್ಟ್ ಆಫ್ ಆಲ್ ವಿಚ್ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್, ಜಸ್ಟ್ ಬಿಫೋರ್ ಐ ರಿಟರ್ನ್ ವಾಟ್ ದ ನೇಮ್? ಅಡಿನೋಸಿನ್ ಆಲ್ರೆಡಿ ಒನ್ ಆಫ್ ದಿ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಇಸ್ ದೇರ್. ನೆಕ್ಸ್ಟ್ ಒನ್ ಇನ್ ದ ಗುವಾನೋಸಿನ್, ನೆಕ್ಸ್ಟ್ ಒನ್ ಇನ್ ದ ಥೈಮಿಡಿನ್, ನೆಕ್ಸ್ಟ್ ಒನ್ ಈಸ್ ಕಾಲ್ಡ್ ಥೈಮಿಡಿನ್ ಅಂಡ್ ಲಾಸ್ಟ್ ಒನ್ ಈಸ್ ಕಾಲ್ಡ್ ಯೂರಿಡಿನ್. ಇದಕ್ಕೆ ಫಾಸ್ಫೇಟ್ ಅಡಿಷನ್ ಮಾಡ್ತಾ ಇದ್ರ. ಓಕೆ, ಈಗ ಹೇಳಿ the phosphate group is attached to the which carbon of the sugar? Phosphate is attached to the which carbon of the sugar? Which carbon of the sugar? Phosphate group is attached to the which carbon of the sugar? Andre which carbon? Fifth carbon. You see, this is already sugar plus base is already there. Andre this is called nucleoside anta kariboda. This is called nucleoside. ಅರ್ಥ ಆಗ್ತಿದ್ರು ಸ್ಟೂಡೆಂಟ್? ಶುಗರ್ ಪ್ಲಸ್ ಬೇಸ್ ಇಸ್ ದೇ, ದಟ್ಸ್ ವೈ ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್. ಈ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ಗೆ ಏನು ಅಟ್ಯಾಚ್ ಮಾಡ್ಬೋದ್ರ? ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಫಾಸ್ಫೇಟ್ ಗ್ರೂಪ್. ಆಲ್ರೆಡಿ ಸೆಡ್ ದಟ್ ಒನ್. ವಾಟ್ ಇಸ್ ದ ಫಾಸ್ಫೇಟ್ ಗ್ರೂಪ್? ಓಕೆನಾ? P, this about the double bond, OH and OH. Come on, already here also OH is present, students. Okay na, CH2OH already is there. Now here also ಏನಾಗ್ತಾ ಇದೆ ಗೊತ್ತಾ ಸ್ಟೂಡೆಂಟ್ಸ್? ಓಕೆ ನಾ? one water molecule removal ಆಗ್ತಾ ಇದೆ. One water molecule removal ಆಗಿ, come on, one of the bond is formation is occurs. What is the name of the bond? Phosphoester bond. This is generally calling as phosphoester bond. Okay, so that's why nucleoside plus phosphate we can call nucleotide anta kariತೀವಿ. Come on, now you have to speak out. Here only you have to write what is a nucleotide. Adenine plus phosphate, what we can call? Adenosine monophosphate anta karibodu. Or it is also called which one? Adenylic acid. Adenosine monophosphate, or it is also called adenylic acid. Because of the presence of the phosphate group and these are having the which nature? Acidic nature. ಅದಕ್ಕೆ ಅಡಿನೋಸಿನ್ ಮೋನೋಫಾಸ್ಫೇಟ್ ಅಂತ ಕರಿಬಹುದು. ಯಾಕೆಂದರೆ ಅಡಿನೋಸಿನ್ಗೆ how many phosphate addition ಆಯಿತು? 1 phosphate. ಅದಕ್ಕೆ ಅಡಿನೋಸಿನ್ ಮೋನೋಫಾಸ್ಫೇಟ್. ಅದಕ್ಕೆ it is called nucleotide. Next one, guanine plus phosphate. ಅಂದರೆ what we can call? Guanosine monophosphate. Or it is also called which one? Guanylic acid. Again, next one. ಥೈಮಿಡಿನ್ಗೆ ಫಾಸ್ಫೇಟ್ ಅಡಿಷನ್ ಆಯಿತು, ವಾಟ್ ವಿ ಕ್ಯಾನ್ ಕಾಲ್ ಥೈಮಿಡಿನ್ ಮೋನೋಫಾಸ್ಫೇಟ್ ಅಂತ ಕರಿತೀರಾ. ಥೈಮಿಡಿನ್ ಮೋನೋಫಾಸ್ಫೇಟ್ ಆರ್ ಥೈಮಿಡೈಲಿಕ್ ಆ್ಯಸಿಡ್ ಅಂತ ಕರಿತೀರಾ. ಥೈಮಿಡೈಲಿಕ್ ಆ್ಯಸಿಡ್, ಥೈಮಿಡೈಲಿಕ್ ಆ್ಯಸಿಡ್, ಥೈಮಿಡೈಲಿಕ್ ಆ್ಯಸಿಡ್. ಈಗ ಲಾಸ್ಟ್ ಒನ್ ಈಸ್ ಕಾಲ್ ದ ಮ್ಯಾಸ್, ಓಕೆ. ಆ್ಯಂಡ್ ಸೈಟಿಡಿನ್ ಅಂತ ಬರಬೇಕಲ್ಲ, ಸೈಟಿಡಿನ್. ಸೈಟಿಡಿನ್ಗೆ ಒಂದು ಫಾಸ್ಫೇಟ್ ಅಡಿಷನ್ ಆಯ್ತಲ್ಲ, ವಾಟ್ ವಿ ಕ್ಯಾನ್ ಕಾಲ್ ದ cytidine monophosphate, cytidine monophosphate, or it is also called cytidylic acid. The last one is also there, it is present only in the RNA, that is called uridylic acid or uridine monophosphate, uridine monophosphate, or it is also called uridylic acid, uridylic acid. Okay students, okay. Idura different type of nucleotides, 4 different type of nucleotides is we can observe in the DNA, but instead of the thymidine, okay now, so uridine is present in the RNA, you already know. Are you getting, boys? Are you getting that one? What is the nucleotides? Okay, so everything, okay na. ಈಗ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಮತ್ತು ಫಾಸ್ಫೇಟಿಕ್ ಮಧ್ಯೆ ಕಮಾನ್ ಒನ್ ಆಫ್ ದಿ ಬಾಂಡ್ ಈಸ್ ಪ್ರೆಸೆಂಟ್. ವಾಟ್ ಇಸ್ ದ ನೇಮ್ ಆಫ್ ದಿ ಬಾಂಡ್? ಫಾಸ್ಫೋಎಸ್ಟರ್ ಬಾಂಡ್. ಫಾಸ್ಫೋಎಸ್ಟರ್ ಬಾಂಡ್. ಫಾಸ್ಫೋಎಸ್ಟರ್ ಬಾಂಡ್. ಫಾಸ್ಫೋಎಸ್ಟರ್ ಬಾಂಡ್ ಈಸ್ ಕಂಪ್ಲೀಟೆಡ್ ಸ್ಟೂಡೆಂಟ್ಸ್. ಓಕೆ, ಸೊ ಇದು DNA ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಬಗ್ಗೆ ಇಲ್ಲಿವರೆಗೂ ಕಂಪ್ಲೀಟ್ ಆಯಿತು. Got the clarity students? Nucleic acid structure students. Okay? Only. ಇದರಲ್ಲಿ ಏನಾದರೂ ಡೌಟ್ ಇದೆಯಾ? ಯಾಕೆಂದರೆ in the same class itself only we have to clarify the doubt. Then only your reading will be continuously occurs without any disturbance. Yes. Got the clarity students? Nucleoside andre adenosine, guanosine, thymidine, cytidine, uridine. Nucleotides andre which one da? Adenosine monophosphate. One phosphate will be addition to that one, the nucleoside. Then or it is also called adenylate acid, like that one it is there. Guanosine monophosphate or guanylic acid, cytidine monophosphate, thymidine monophosphate or thymidylic acid, cytidine monophosphate or cytidylic acid, uridine monophosphate or uridylic acid. Confuse agಬೇಡಿ, sometimes uridylic acid ಅಂತ ಕೊಡ್ತಾರೆ or sometimes thymidylic acid ಅಂತ ಕೊಡ್ತಾರೆ. So that's why thymidylic acid ಅಂದರೆ thymidine monophosphate. Okay na? So phosphoester bonds are present. Phosphoester bonds are present. This is about ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್. ಅಂದರೆ ಈಗ ಗೊತ್ತಾಯ್ತಪ್ಪ, ಅಂದರೆ ಶುಗರ್ ಮಾಲಿಕ್ಯೂಲ್ ಎಟ್ ಒನ್ ಸೈಡ್ ಇಟ್ ಈಸ್ ಅಟ್ಯಾಚ್ಡ್ ವಿತ್ ಹೂಮ್? ಶುಗರ್ ಮಾಲಿಕ್ಯೂಲ್ ಎಟ್ ಒನ್ ಸೈಡ್ ದೇ ಆರ್ ಅಟ್ಯಾಚ್ಡ್ ವಿತ್ ಹೂಮ್? Nitrogenous base, and another side they can attach to whom? Phosphate group, correct? Then only they will form the nucleotide. Nucleotide under 3 components bartara. What are the 3 components? Nitrogenous base, sugar and phosphate. So nitrogenous base, sugar, both are together what we can call nucleoside anta karitara. Iga nucleoside plus phosphate, nucleoside and phosphates together what we can call nucleotide anta karitara. Idaralli phosphoester bond is present here. Which one andra? Glycosidic bond is present. A glycosidic bond is present, students. Clear? Okay, next class we can discuss about binucleotide. Do you know what is meant by binucleotide? Yes, 2 nucleotides, if they will completely bind with each other, what we can call binucleotide. What is meant by polynucleotide? Many nucleotides, correct? More than 2 nucleotides, if they can bind with each other, that is called polynucleotide. Okay na? So binucleotide, polynucleotide also barbeku, but with atomic structure I can draw. ಯಾಕಂದರೆ ಅಟಾಮಿಕ್ ಸ್ಟ್ರಕ್ಚರ್ನಲ್ಲಿ ನಿಮಗೆ ಎಲ್ಲ ಕಂಪ್ಲೀಟ್ ಗೊತ್ತಾಗಿರ್ಬೇಕು ಸರ. ಇದು ಓನ್ಲಿ ಎಸ್ಟರ್ ಬಾಂಡ್ ಅಂತ ಕರೀತಾರೆ. ಅದೇ ಒನ್ ಮೋರ್ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಬರುತ್ತೆ ಅಂದರೆ ಅದನ್ನು ಎಸ್ಟರ್ ಬಾಂಡ್ ಅಂತ ಕರೀತೀರಾ, ಫಾಸ್ಫೋ ಡೈಎಸ್ಟರ್ ಬಾಂಡ್ ಅಂತ ಕರೀತಾರೆ. ಅದಕ್ಕೆ 2 ಎಸ್ಟರ್ ಬಾಂಡ್ಸ್ ಆಗಿದೆ. ಯಾಕೆಂದರೆ ಆಲ್ರೆಡಿ 1 ಎಸ್ಟರ್ ಬಾಂಡ್ ಇದ್ದೀರಾ, ಮತ್ತೆ ಇದೇ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಇನ್ನೊಂದು ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಜೊತೆಗೆ ಬೈಂಡ್ ಆವಾಗ ಇನ್ನೊಂದು ಎಸ್ಟರ್ ಬಾಂಡ್ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ. ದಟ್ಸ್ ವೈ ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಫಾಸ್ಫೋ ಡೈಎಸ್ಟರ್ ಬಾಂಡ್. ಓಕೆ, always nitrogenous base are attached to the which carbon of this sugar? First carbon of the sugar or right side of the sugar only it is present. Idu tilkoli students. First carbon andre mainly right side of the sugar only nitrogenous base are attached to sugar molecules.